the height of the COVID-19 pandemic in 2020 in Qatar, Noor volunteered at the Communicable Disease Center as a volunteer lab technologist, where she aided in processing COVID-19 swab samples by RNA extraction methods. Later in 2022, she had an opportunity to attend the Medi-Euro Network Workshop, Biobanking and Biomedical Research in Cyprus, and she shared her knowledge and experience working in a biobank and learned about biobanking in different countries and their different approaches in achieving and establishing a biobank. As of 2018 and to date, Noor is a laboratory technologist at Qatar Biobank, Qatar Foundation, where she performs processing and storage of different biological sample types, DNA extraction and quantification, and running the automated BioStore for sample storage. Qatar Biobank Lab has achieved College of American Pathologists accreditation in 2020, as I mentioned, and accreditation in 2022, which Noor is a part of the team that worked on achieving these milestones. So that being said, I just want to give a great welcome to Iman, Monica, and Noor for today's Biobank Showcase. Welcome.
Thank you.
Good morning, everyone. Thank you for joining this session. And I would like to thank the IHCC organization to give us this opportunity to take you through our journey in how to build and implement Qatar Biobank Laboratory. Our presenter will be Iman, Monica, Noor. The presenter have no conflict of interest or commercial base to declare in relation to this presentation. In the first part, we will speak about establishing a biobank laboratory, planning and implementation. The presenter will be me, Iman Al-Khayyat. I am as a lab manager, and my colleague, Noor Hamad. Our objective: identify key requirements for establishing a biobanking laboratory, understand the 12 quality system essential as parallels of successful laboratory organization. We will introduce the laboratory scope, covering pre-analytical, analytical, and post-analytical requirements. Then, we will demonstrate the advantage and disadvantage of manual and automation of sample processing and storage management. Our content will be introduction, quality management system, specimen collection and the process, lab operation, manual and automated, sample storage, and finally, facility and health and safety. When we start to implement any laboratory or any organization, we need to build strategy. We need to put our scope, our strategic objective, our plan and goals, what the resource required to implement this organization, and what the stakeholders, the internal and external stakeholders. We will start talking into 12 quality system essential to establish and maintain the stability in laboratory. These 12 quality essential system can be considered as a guide when establishing a lab, and we will be highlighting them throughout this presentation. 12 quality system essential are a set of quality standards that provide a framework for maintaining a quality management system in biobanks laboratory. These quality essential systems include aspects such as management responsibility, document control, personnel training, equipment management, process control, and customer service. Each of them is essential to ensure accurate, reliable, and consistent result in biobank laboratory operations. Now we will speak about the facility and infrastructure requirement. I handle this part to my colleague Noor.
Hello, everyone. Thank you again for giving us this opportunity to speak today. Our first quality system essential will be discussing facilities and infrastructure requirements. The three main resources to take into consideration when establishing a biobank lab are financial, facilities, and personnel. A budget should be put into place taking into consideration the number of personnel needed, inventory required, and equipment. Facilities should be adequate depending on the processes being performed. There should be enough space, properly designed and constructed rooms, and proper environmental control measures. Thirdly, personnel working in the lab should be competent and able to perform the tasks assigned. As mentioned, facilities should be adequate depending on the lab processes. Some space requirement examples are: workstations should be spacious enough for work being performed, meet ergonomic standards, strong enough to hold equipment. An office space should be provided, which can also be considered as a clean area, meaning no samples or contaminated equipment allowed inside, for staff to do office and admin work related in the lab. UPS and generators are important as they are a backup power supply in case of power failure, and rooms requiring electrical supply should have UPS outlets installed. As I mentioned, environmental control is an aspect to be considered when designing a lab. It is important that the lab workstations are kept clean at all times. This can be achieved by establishing workspace decontamination procedure, as well as routine housekeeping. Next, biological cabinets and negative pressure rooms can be implemented if samples and processes require so. If automation and instruments are used, the noise level, electrical testing, waste management should all be factored in and controlled during the processes. Rooms should be temperature controlled and monitored for temperature-dependent equipment and temperature-dependent samples. The lab requires many equipment and consumables, reagents. These should all be housed in a proper way to not cause any clutter. Therefore, a designated storeroom should be another consideration in the lab. This storeroom is also temperature and humidity maintained to not spoil any inventory or reagents. Within the storeroom, adequate shelving and cabinet spaces are useful to organize the item, making them easy to see and easy to access. Flammable reagents should be separated from non-flammable reagents. For reagents that are temperature sensitive, storage fridges and freezers can be used, as well as safety cabinets for flammable reagents. The lab should be considered as a restricted area, meaning only authorized personnel are allowed inside. As well as the lab rooms, the storage room and sample storage units should have restricted access. This access could be achieved through access cards and keys assigned only to authorized personnel. Another quality system essential is information management. It is one of the most important pillars of successful process implementation. By equipment integration to a LIMS system or a laboratory integrated management system that leads to enhanced sample traceability, as well as confidentiality and integrity of our data following ISMS policy. These LIMS systems allow for sample data viewing and modifications. Our next section to discuss will be regarding health and safety in the lab. There are basic health and safety measures that are important to be implemented. These include personal protective equipment that should be provided to all staff, as well as training on how to don and dope the PPE properly. First aid kits should be accessible and available. Biological and chemical spill kits, along with training on how to use them. Eye wash stations and showers should be implemented in proper areas, as well as training on how to use them. MSDS and SDS to be provided for all the chemicals in the lab. Finally, fire safety equipment to be available and training to be provided, as well as fire drills. Some non-physical aspects to consider regarding health and safety in a lab. Establishing a lab safety committee. They will meet up to discuss and provide updates on any health and safety issues and concerns. It's also a good idea to have internal lab safety audits performed on a periodic basis. Staff to follow good lab practices for continuous improvements. Trainings to be given to new staff, as well as refresher trainings to be given to existing staff. Since a lab generates different types of waste depending on the process to be performed, the waste generated should be handled and disposed of correctly as not to pose any health risk to lab personnel as well as the general public. Some common types of waste generated are biological, liquid, general, sharps, and chemical. Regardless of the type, waste should be collected in a designated and controlled area, and waste should be labeled and tagged correctly. Waste should also comply with a country's waste management and disposal laws. I will now hand over the remaining of the session to Ms. Iman.
In this session, we will focus on the sample collection and the processing. There are multi-studies nested in Qatar Biobank. Each study requires specific design kit to meet the requirements. And when the kit is created in the lab, each kit has specific number linked to the participant. And these kits collected by our nurses from lab for withdrawal blood extraction from participant. There is an order for extraction the blood to avoid the additive cross-contamination. We are focused on the healthy participant and some infected patient. We collected different blood tube, urine and saliva from each participant based on the design of the kit, and maximum volume of blood is 60 ml. When the blood is collected and received to the lab, there is distribution of the sample. Some of the sample will go inside Qatar Biobank laboratory for initial fractionation of the sample, and the remaining of the tube, like 6 tube, 3 EDTA and 2 gel tube and 1 sodium citrate for biomarker analysis. As we are unique in our biobank, we will offer a full checkup, including multiple analysis and analysis shipped for more than 70 blood biomarker, which this blood tube shipped to third-party governmental hospital. And when we received the data from governmental hospital, the medical team will provide the participant with the feedback on their result and referral if they needed in this stage. In the lab operation, our scope in the Qatar Biobank laboratory in the beginning as limited, as we started by manual fractionation. Then we will expand this scope in 2016 by start the first automation, which is easy blood fractionation, which was integrated with iLEMS. In 2017, we implemented further automation, like DNA extraction based on the magnetic bead protocol and simple process for measure the quantification and concentration and the purity. In 2018, we implemented two automation. One automation for RNA extraction based on the magnetic bead protocol. The other one is automation for quality control, such as SNP trace panel, to check the quality of DNA and cross-checking that the genotyping data is cross-binding with the phenotyping data. And finally, in 2019, we introduced our microarray, or Qatari chip, to detect any changes in DNA and discover any disease if infected by in the Qatari population in the future. We have seven laboratories in Qatar Biobank, or 7 process: sample receiving, sample processing, silver preparation tube, DNA extraction, RNA extraction, microarray, and finally the quality lab. There are quality control in any laboratory in the world. There is some pre-analytical, analytical, and post-analytical. In the pre-analytical phase, in this stage we have in the sample receiving and sample processing. In the sample receiving, there is some quality check. Like when we receive the sample, we should check the quantity of the sample and condition of the sample if there is any clot or hemolysis. Also, the transfer of the collected sample, or the time from collected until we receive it to our laboratory, should be within 15 minutes. And the transport of other non-clinical sample for the 70 biomarker analysis for governmental hospital should be with controlled temperature from 2 to 8 and within 4 hours. In the sample processing, our quality check should be processing within 2 hours as maximum, include centrifuging time and speed, and serum clotting time. In the analytical phase, we have some also quality check, like we have as our process as initial fractionation, automated or manual. We needed some quality. To measure the white blood cell count, we use hematology analyzer for cell count, and also for behavioral blood mononuclear cell, we use it hematology analyzer for cell blood count, and also other method which is the flow cytometer to measure the cell viability. Then, to detect the volume is dispensed in the machine in the accurate way, or fractionation by personnel in the accurate way, we have volume detector to detect the volume. Also, there is analytical phases for other process in the lab, like DNA and RNA extraction. There is DNA quantification, we use it fluorometric process. And for purity, we measure by spectrophotometer process. And for DNA integrity, for genomic score, we use it microfluid process. And each type of the sample, DNA and RNA, have different range. And this is quality considered as analytical QC. There is functional quality QC only for DNA type. DNA fingerprinting for sample traceability for sample swab, mix up gender confirmation with demographic data. And in the microarray process, we have 3 different microarray in our Qatar Biobank laboratory. And we have some quality check, like a plate pass rate should be more than 95%, and the QC core rate of passing sample should be more than 98.5. And finally, what are the post-analytical phase in the quality check? We have the post-analytical in 2 processes, in the sample storage process and in the retrieval and distribution process. The sample storage process, we use the sample inventory validation to manually check if the sample in the correct location, and my colleague Monica will speak more in details in this part. For retrieval and distribution, when we provide the researcher with the sample, we should prepare in the same time the quality report with all relevant information. The manual process. What the infrastructure required in the manual process? The workplace approved by Ministry of Public Health, constant room temperature. What the equipment required? Required simple equipment like pipette, tips, centrifuge, biological safety cabinet, microtubes, freezers, fridge, bench or table, and plate, and caps and secure. What the advantage and disadvantage from this process? The advantage is low cost, processing all material without complex software program, easily handling. We have limited number of aliquot in that time. We collected 73 from each participant. What the disadvantage? There is many human error. No sample tracking system, that means no traceability. Low sample quality. High number of collected sample to be processed need high number of personnel. And sample distribution plate, we have 4 plate. The first 3 plate are similar, but the 4th plate have different type of the sample, like buffy coat, urine, and saliva, and this is not replicated in other plate and can be lost at any time, as this is the only one copy. For automated sample process, as I mentioned, and the automation part, the advantage we can higher throughput, and we have good quality of sample. There is a traceability. There is organized sample distribution plate map, as we have 3 copies of a plate, and all plates are similar. The disadvantage is high cost. The backup required in case of any failure of the machine. The first quality system essential is equipment and its management. Where we need to take care of various aspects such as installation. Before the installation, we should put the spec of the machine, then send it to procurement. Then, after that, we received the machine, we needed to make the installation by the supplier, validation, installation by manufacturer or the supplier, validation, maintenance, calibration, troubleshooting by the staff is required, repair and maintenance and service report if any things happen in the machine, and records for everything like checklists and records for troubleshooting by the action or accidents happen in the machine. Following should be considered before purchasing the device. Quality essential for establishing automated process, including device qualification. As I mentioned in the previous, we need to put the spec, installation qualification, operation qualification, and performance qualification. These will be discussed in the details by our colleague Monica in next part of the presentation. All equipment used should be documented. Our equipment maintenance is tracked through master list of equipment. For successful day-to-day operation, it is essential to have good documentation, easily accessible for all lab staff, and it has to reflect the last changes and the flow chain of approvals, revision, review, distribution, archiving, retention time should be documented. Here are some basic documents should be considered when establishing a biobank laboratory or any lab. Policy and procedure, standard operator procedure or work instruction, health and safety program, chemical hygiene plan, risk assessment, business contingency plan, maintenance log sheet, checklist, procedure to address non-conformities, sample distribution, participant withdrawal procedure. Another quality essential system, purchasing and inventory, where we have developed work instruction to be able to have systematic approach to maintain our supplies and inventory management. And these included in this document, we included the vendor qualification, supplies and reagent, critical service, contract review, inventory management. And we should consider when we do any inventory, our conception, clearance, expiry date of the reagent, shipment time, stability, license, dangerous good. Inventory tracking and monitoring is important to not face any shortage of expiry or expiry reagent. In the lab, we started off with a simple Excel sheet where we manual input information of the item received, and on monthly basis, we would count the inventory and document it on this sheet. Then, the IT department make it our life easy. They implemented for lab some system, in-house system, which is make it easy tracking and searching for item and monthly updated of the same. Also, in this system, there is privilege as there is restriction on who can edit the inventory and alerts for inventory running low and nearing expiry. Organization and leadership provide a framework for creating a culture of equality and continuous improvement. This needed human resource, job qualification and job description by different skills, as we have different tasks in the lab is required by different personnel, like we need to structure the org structure, to build our org structure, to include lab technologist, lab technician, lab manager, safety officer, quality officer, and each of them have different tasks than other. A systematic approach to managing and improving laboratory operation by identify and addressing unexpected events or accident by occurrence management. And this contain complaints, mistake and problem, documentation, root cause analysis, immediate actions, corrective actions, preventive actions. QSE, process improvement, and customer service can help to improve the workflow, reduce errors, and enhance the quality of the result. Additionally, by prioritizing customer service, laboratories can enhance communication with the clients and provide high-quality service that meets or exceeds their expectation, in our case, with our stakeholders. In this process, we have opportunity for improvement. We have stakeholder feedback. We have a problem resolution. For sample storage, we have manual process, which was manual storage. We need simple freezers like chest, upright freezers, and liquid nitrogen tank. There is a room temperature should be maintained as required. In that time of the building of Biobank until 2015, 2016, we have Excel sheet to track the storage location. We have backup from the time of building a biobank laboratory. We have remote site location as a backup for any accident. What the advantage from manual storage? Low cost, not too high technology and complicated system. Any type of labware can be stored, but the disadvantage, not easily to access to all samples. Freezer temperature fluctuation, it's happening through the retrieval process. No sample tracking as we use in that time, as I mentioned in the previous slide, Excel sheet. About the automated storage. We implemented our automated storage in 2018. There is different type of micro tube and different size. We have -80 storage zone and -20 handling zone, 2 step of refrigerating, input, output. The capacity of automated store 2,300,000 sample. The backup will be secondary refrigerating and liquid nitrogen in this automated biostore, in the automated store in our laboratory. What the advantage? The advantage improved the sample quality, data directly stored in database, no constant staff intervention. The disadvantage, temperature control, humidity, costly to program and reprogram, space requirement. For sample distribution plan, as I mentioned, from the advantage of automated, we have high throughput. We divided the sample into 3 copies according to different storage location. We have automated -80 in our facility and the cryopreservation, which contains 7 liquid nitrogen tank in our facility. And we have also remote site or backup outside the biobank, which we keep the second copy of the sample there to avoid any disaster. But according to some size of the sample, we cannot storage in the automated store. We should keep it 1 copy, like for example, in case of Buffycode and Bexgene for extraction process, we keep it 1 copy for extraction and other copy we store it in our backup in remote site. and this is referred to ISPR Best Practice, 3rd edition, 2012. After implementation of our automation, we are able to increase our quality plan to produce more samples as we have high throughput. From 73, we reached in 2016 114 fractionation samples from each participant. But later, regarding to reduce the demand of some type of sample from the research, we reduce number of some sample, and in the same time we increased our capacity. And the QBB laboratory has worked on overcoming and challenges of sample storage validation as no reference standard or guidelines with formula to calculate the inventory sample storage validation. The inventory storage validation has been designed to cover systematically all sample storage condition, sample type, and the storage location based on defined percentage 0.1%. Now, there is a video to take you through QBB laboratory. Enjoy in this video.
Welcome to Qatar Biobank. Qatar Biobank was launched in 2012 as part of a collaboration with the Ministry of Public Health and the Hamad Medical Corporation. Through the collection of samples and information on the health and lifestyles of Qatari citizens and long-term residents, Qatar Biobank aims to make vital medical research possible for scientists in Qatar, the region, and the world. Qatar, like many places in the world, is plagued with complex health challenges such as heart disease, diabetes, obesity, and many types of cancer. We have created a proven mechanism that makes it possible to perform research utilizing new data in hopes of solving these challenging health problems. Our state-of-the-art biobank facility has drawn national and international acclaim as a premier biomedical research facility in the region. Our processes harness the most advanced technology, allowing us to advance and consistently deliver quality results quickly. Our highly educated staff, advanced technology and operational philosophy ensure the highest level of ethical and quality standards. The QBB utilizes the gold standard in laboratory equipment and techniques. Let me introduce you to some of this incredible technology, including a processing lab, a CPT lab, a flow lab, and a DNA lab, in addition to the storage facilities. The samples are received here at the reception desk. The process begins with every sample being marked with a unique barcode. By generating the barcode at the time of sample collection, every sample is tracked through all the different processes and methods performed at the biobank. Some of our most sophisticated instruments automate and optimize our lab processes, and these include blood fractionation workstations, cell blood counters, and nucleic acid extraction workstations. These instruments perform at a level and are unparalleled in precision and efficiency. The blood fractionation workstation allows for fully automated blood fractionation and buffy coat isolation and is located within Qatar Biobank Processing Lab. This device is precisely engineered to support the sample preparation process and provides safety for sample integrity, its associated data, as well as the donor and operator. This system is fully automated, efficient, and allows sample tracking through the entire process. Viable cells extraction occurs in our biological safety cabinet in our CPT lab, which is designed to protect the sample, worker, and environment. Our flow lab conducts viability testing using various state-of-the-art equipment, including flow cytometry instruments. The cell counter is a precise quality control instrument we utilize for performing blood counts. This device delivers the ultimate in precision cell counts and is automated, efficient, and cost-effective. Isolation of nucleic acids is performed using state-of-the-art technology. to ensure the best nucleic acid purification results for all downstream processes. This high-throughput device can handle a variety of sample sizes at relatively high speed, allowing the quick processing of samples in a single day. Our system gives you the flexibility to analyze samples immediately or safely store for later use. You will find our cryogenic and automated storage systems are state-of-the-art, with the capacity to store over 2,300,000 samples. Sample integrity is critical throughout all phases of the sample process, from collection to processing, storage and delivery to the researcher. Our storage systems maintain the integrity of the stored sample for future use. Qatar Biobank's storage facilities are secure and comply with international biological storage and safety standards using proven fail-safe measures, which include rigorous compliance and monitoring systems, along with O2 level detectors. We pride ourselves on our commitment to quality and information security. Qatar Biobank is ISO accredited for QMS 9001-2015 quality and ISO 27001-2013 information security management, which provides assurance that our procedures are standardized and validated, and the quality of our samples and data collected are consistently high. We have set the standard for biospecimen and clinical data collection, allowing researchers to imagine a better future of personalized medicine through the work of Qatar Biobank. We warmly invite you to visit us today.
Thank you. The Qatar Biobank team I believe this is the end of part 1. Please feel free to put your questions in the chat. I'm not seeing any questions as of right now, but I'll give you all a few moments to please put your questions in the chat. All right, I see a question from Vanessa. She said, thank you for a very interesting presentation. Could you please explain how the automated sample storage system works? And how do you decide when to use freezers and when liquid nitrogen tanks? Could you please explain your sample retrieval process? So I'll hand it over to Iman, Noor, and Monica to answer this question, or these series of questions.
Hello, thank you for your question, Vanessa. So addressing the first point about our automated sample storage, it's a two-step refrigeration system. So the sample, either if they're stored in the fridge throughout the day or if they've freshly processed at room temperature, when input in the biostore, they will reach up to -40 degrees Celsius. And then the second step is from -40 to -80 degrees Celsius. And that's the final storage temperature. For the second question, how do we decide when to use freezers and liquid nitrogen? So as Ms. Iman mentioned in her slide, each sample is processed into 3 copies. 1 copy goes to the automated biostore, 1 will go to the liquid nitrogen tank, and 1 will go to the remote site, which is a location outside of Qatar Biobank building. And the liquid nitrogen for us is our long-term storage. So in any case where automation might fail or we cannot input our samples in the automated biostore, in Biobank, we do have backup freezers of -80 degrees where we store the samples temporarily until we are able to input them in the biostore. And regarding our retrieval process, if there is any request for a research, it will go through our IT department. We will approve the samples that meet the requirements. And through a pick list, we will generate what we call a pick list in the biostore, where there's a list of the sample aliquots that are needed. and the BioStore will retrieve them accordingly, according to their barcode and their storage location. And after the pick order is done, we will get an alert and we will be able to output these samples.
Thank you for that response. Is there any other questions? I see a question from Dr. Walid. Are you storing samples based on diseases? If yes, what such diseases do you have? Are you able to see the question from Dr. Walid? Dr. Walid asked, are you storing samples based on diseases? If yes, what such diseases do you have?
Okay. Hello, Dr. Walid. Thank you for your question. So in general, we do work on a healthy population. So all of our sample go to the three storage distribution plan, as I mentioned earlier. But during the pandemic, when we were collecting samples from COVID positive patients, we did have special allocated freezers where we only dedicated them for our COVID sample storage. Regarding follow-up samples later on from the follow-up patients after COVID recovery, we continued storing them in our automated BioStore, our liquid nitrogen, and our remote site, just as the healthy participants. So we don't have any diseases or any specific diseases or any specific storage for any diseases.
I see Latifa has her hand up, and Latifa, do you want to come off mute and ask your question?
Yes, thank you, Chisholm. Thank you, Iman and Noor, for this great presentation. Yeah, so I have just a general question about lesson learned. So which, if you can name or list a few of the lesson learned that you can share with the audience, especially for the biobankers who are preparing to get accredited. Thank you. Iman, Noor, did you get my question or you want me to repeat?
Yeah, can you repeat it, please?
Okay, yeah, I'm just asking if there are any lesson learned that you would like to share with the audience, especially for biobankers who are preparing for accreditation. Shisa, maybe you can see there are other maybe question or something in case they
Yeah,
need
I'm seeing
to
the
respond.
question in the chat. Iman, Noor, and Monica, are there any lessons learned that you received through the accreditation process that will be useful for other people that are biobanking? I think that's the question we have currently.
If you ask about the accreditation, we learn from each standard for each accreditation how to create the document related to this standards. This is a question, you mean it, or you mean something different? Because your question is not clear.
Okay, no problem. We can I mean, move on to other questions because I don't want to take too much time. I'll follow up with you on the lesson learned later on, then I can share them with the audience if needed. Okay, thank you.
All right, so I see in the chat, we have another question from Vanessa and then Senthil. So Vanessa asks, how do you determine the maximum volume of blood that can be collected?
Based on our consent, we have the maximum of blood can be extracted from the participant.
All right, and then the last question is, do you have systems on how you capture the samples dispensation? And then we're going to take a break after this.
Like 5 minutes break, Chisam?
Yeah, 5-minute break. But actually, what we can do, are you able, Iman and Noor, are you able to answer the question in the chat?
Yeah. About the last question about how the eye limbs capture the sample dispensation. Yeah, our eye limbs capture the sample dispensation as our automation have camera, and this camera based on the color to fractionation of the sample. And we have picture for each fractionation for each type of the sample and kept in our database.
Thank you for the questions. Thank you for those responses. We're going to take a 5-minute break and come back about 3 minutes after the top of the hour. Thank you. All right, thank you everyone for this break. Monica, you can start your presentation now. I'm not hearing any sound from the presentation. Has it started?
Sorry? Okay, sorry. Can you repeat? You cannot hear me?
You cannot hear the presentation.
Okay, just a second.
I think you might have to unmute yourself so that we can hear it coming from your computer. So if you unmute and then click play, let's see if that works. Yeah, we're still not hearing any sound.
Can you hear it now?
Nope. Maybe if the previous presenter presented from their screen.
Okay, let's just start. Good morning or good afternoon. I'm Monica, and before I start, I just want to take this opportunity to thank you 1000K cohort for organizing this webinar and giving me the opportunity to showcase Qatar Biobank laboratory quality standards, journey and implementation. The presenter has no conflict of interest or commercial bias to declare in relation to this presentation. Objectives for today's presentation will cover: list the benefits of accreditation and quality standards in laboratory management, discuss the significance of quality assurance and quality control in routine laboratory practice, distinguish the process validation from process verification and list the parameters. Our content for today will start from the introduction of the concept of standard, guideline and regulation. Then we will show you our certifications and accreditation and how we implement it and how we are monitoring quality control and quality assurance in our laboratory. And we will end with fundamentals of laboratory process and method validation and verification. As a biobank laboratory, we collect, store, distribute biological specimens for research and diagnostic purposes. It is crucial that we maintain the highest standards of quality in our work to ensure simply the accurate and reliable results. To achieve this, we need to follow the set of standards and guidelines. This structure of quality management system you had a chance to see in previous presentation presented by my colleagues, Ms. Iman and Ms. Noor. For the quality standards I am going to focus today, it will be our quality implementation and quality fundamentals needed to run a successful laboratory. But before we move to the detailed explanation, I would like to introduce 5 terminologies often used in relation to quality. Regulation, it's a regulatory standard written by a national government. Standard, there are requirements for regulation of the processes and they are recorded in the documents. It's basically telling us what to do. Guideline, it's describing how to implement the standard requirements. So it's telling us how to do it, how to apply the standard. Accreditation is a process which then authoritative organization give a formal recognition to the laboratory that it's competent to carry out specific tasks.
And certification, it's official recognition that an organization has met a specific quality standards and criteria. In this table, you can see what are the most common standardization and international bodies. So ISO is the International Organization of Standardization. TLSI is the Clinical and Laboratory Standards Institute. CEN is the European Committee for Standardization. WHO is the World Health Organization. So all of them are providing standards and guidelines. So, but they are a bit different. Here you can see the two major models for quality management system used globally. So the ISO, which is very broad-based, and TLSI is very specific based. So there is set of the standards and guidelines and best practices, and they are tailored specifically for the medical lab testing. And combining these two was really challenging for us, but in the same time, we gained from both of the QMS standards in a way that we really improved our processes. In the next in the presented table, you can observe the classification of quality documents and decide which one can fit to your own laboratory scope and to be tailored to your own lab specific needs. Is it clinical or not clinical? And is it international or national standard? So here it's a really good comprehensive guideline for anyone who is just starting and it's getting confused with a lot of requirements. So here you can see who is developing and who is maintaining these standards and guidelines. Our Qatar Biobank quality journey started back in 2012, where we achieved our first quality management system and information management system ISO accreditation certification at 2014. And every subsequent year, we were successful in expanding our scope. And at 2020, we got a CAP accreditation, the most stringent and prestigious accreditation for diagnostic laboratories. We achieved it for our in-house developed custom-designed microarray. However, we are implementing all the requirements to our full biobanking scope to maintain the same quality of the samples, considering the Qatar Vision 2030 for precision medicine implementation. So why CAP accreditation and CAP inspections? So here, the benefit for the laboratory is mostly about the laboratory improvement, and the main focus is a patient and the better patient care. In the same time, there is always ongoing compliance with the regulations and with the standards, and it's getting the lab to have an opportunity to stay tuned with the latest requirements. So the checklists are revised every year, and there is more than 2,600 checklist requirements. They are reflecting the best practices, and the CAP accreditation requirements are highly tied to the TLSI guidelines. So that means that they are very specific. So the CAP accreditation cycle is a 2-year cycle where it will be the first external on-site inspection from the CAP expert team, and the following year will be interim CAP inspection, which will be done by the people who are already in the lab. So it's like an interim CAP inspection, and after that, the cycle is repeating every 2 years. As a result of collaboration from a binational working group between the national healthcare provider and the Qatar Biobank, there is a custom-designed microarray, and it was developed. It's called SNP-targeted microarray. We call it QChip. So what you can see here is a DGDQBB CAP activity menu on the CAP website listing which clinical service we are offering to healthcare provider, the biggest governmental hospital in Qatar, as my colleague Vilma mentioned in her previous module. So this is a great example how the collaboration between biobanks and the healthcare provider can result in achieving the highest quality and the highest care for the patient. The other milestone in our quality journey was implementation of ISO 20387 biobanking standard, where we benefited as a lab from improving our specimen handling, storage, and management of quality of the samples and their associated data. We implemented interoperability and we reviewed our ethical practices, and in the same time we got a more insights in how to design our quality controls to achieve the fit for purpose requirement in this standard. As you have seen, our laboratory has to comply to various standards, accreditations, and guidelines, and they all have their own specific requirement for the competence of the biobanks or the biorepositories. As you get that comprehensive overview from the previous part held by my colleagues about the quality system essentials, I just want to complete that journey and to focus on the remaining two quality system essentials. There is a quality essential assessment and a process management. Laboratory assessment is a very important aspect of quality management system in any organization involved in laboratory testing, research, or analysis. And as internal laboratory assessment is an evaluation of the laboratory quality system and operations conducted by trained personnel within the laboratory or the same organizations. So as you can see here, the internal quality controls are something that it's a daily activity and it's performed on routine tests which are present in the lab every day with the same lab personnel, and they're leading to precise results. And in the other way, there is external quality control, which can be a program or it can be the accreditation body who is doing the external quality assessment of the scope of the laboratory. And for the external quality control is specific that it's a periodic and it's a retrospective review of the lab processes. And also it indicates that the laboratory staff areas of improvement or if there is a need to change anything in the quality management system for the quality controls. And at the end, it maintains accuracy. So both of these controls are complementing each other, and the lab can benefit from getting, the precise results, and at the same time, we can maintain our accuracy. So the quality system essential for the laboratory assessment for internal one, there are some of the examples what we have implemented in our lab. So we started to evaluate our reagents, so lot-to-lot comparison. That means that we will receive our reagents and we will compare the positive control run on the previous lot and the new lot. Or if we have a same lot of the reagents, but they come in different shipments because, again, the conditions were different, we are going to compare. We have set up the criteria which we will accept or reject the lot. At the same time, in most of the labs, there is something called instrument-to-instrument comparison or method-to-method comparison. If we are having, for example, automation and a manual process for, let's say, DNA quantification, we want to know that in case we have any downtime for the automation, we can easily perform a manual one, and this will not compromise our results. So that's why we implemented periodic review, periodic comparison with set of acceptance criteria between two methods. And if we are having the same instruments, and we have to make sure that they are interchangeable at any time, because we don't know what can happen or what kind of workload we are going to get. So this is one of the measures that we implemented. And in the same time, because of many requirements from our accreditations and certifications, we have to perform a regular periodic audit programs, audit reviews, quality management reviews, and finally, we can quantify our success by performing and monitoring quality indicators and key performance indicators. For the quality indicators, it's very important to memorize that there are specific criteria for the standards which laboratory must meet to ensure that it's operating at the high level of quality and accuracy all the time. The important characteristics here that these indicators are specific and they are test related and they're measuring accuracy and precision, or they will measure the turnaround time needed for the test completion. And at the end, it's overall efficiency and effectiveness of test method, of the test or implemented test protocol. So here you can see one of the examples that we are measuring. It's called plate metrics. We have a certain criteria that it has to pass 98.5% of all the total samples in that plate. So what we did, we developed a formula, and we set up our criteria, and every quarter we are revisiting our results, we are evaluating and documenting it. And this is going under the management review. And if there is any deviation or any reasons for concern, we will immediately follow our corrective action procedure for accuracy management. And from the other hand, key performance indicators are metrics that are used to assess the overall performance in relation to specific goals or objectives. So what that means? It means that they are not test specific like quality indicators. And the other side, they are giving us idea about overall accuracy and overall reliability of our results. They are easy to identify areas of improvement, and for most of the laboratories, they are used for benchmarking. So what we implemented in our lab, and we took this from the ISPER best practices, is that we are calculating the percentage of the samples aliquoted from the total number of blood tubes received. And we set up our criteria and we are reviewing it quarterly. And this can tell us about our achievements, and we really can identify some areas where we need to improve if that will be the case. When we talk about the external laboratory assessments, the first thing that comes to our mind are proficiency testing. So the proficiency testing is very beneficial for every laboratory because it's ensuring the accuracy and reliability of testing methods. Again, it will identify areas for improvement. It will demonstrate the quality of the laboratory work, because all the results are going to be compared with a big pool of other laboratory results. So we can exactly see where we are standing in comparison to any global network who are performing the same test methods. And this will also lead to exchange of knowledge and expertise in terms of the networking. And in the same time, it's going to support the development of new testing methods, which is always challenging. And this is what's going on rapidly in every laboratory. But it's not always so straightforward. So for some of the tests, especially for the biobanking scope, it's not always available, the official PT provider who is having a PT accreditation. So then the lab has to be creative, and there is something called alternative external quality assessment, which is basically a collaboration between two laboratories. For example, the split sample analysis will be just a simple interlaboratory comparison, but the lab who is providing the samples will be used as a reference lab. And that's why there are some factors that I'm going to show you on the next slide, but it has to be considered when choosing the alternative external quality assessment lab that you are going to work with. But the common for both types of proficiency testing, so is it like from the provider or from the other lab, is important that when we receive the samples, the proficiency testing samples, that we are maintaining them in the same way as any other routine samples. They shouldn't be any warning that this is a PT sample and now we have to get serious and to work closely. No, it should be really treated as any other sample, because in this case, and only in this case, you are going to get the real feedback from your process, and then you can take an action to improve that. So for us, we approached two alternative external quality assessment providers. So for the one, it was a US provider, and we enrolled there from 2019. we were good, so we enrolled in 2 PT rounds per year. And also we enrolled with the local governmental hospital laboratory in the same way, and we are also performing 2 PT rounds per year. The important and the common for both is that they should be CAP accredited, or they should have the same scope and same accreditation that what you are aiming for as a lab. And for the biobanking scope, we approached the IBBL PT provider, and because they have a great proficiency testing programs which are covering a lot from our biobanking scope, and also the frequency is biannual, same as for the other ones. So as you have seen today, the QMS program covers all mentioned 12 the quality management system essentials. So at the last but not least is a process management. So what that means and what it covers. So it means that we need to find the most efficient way to perform and to document all our QC activities. And one of the most important document here is called the QM plan, quality management plan, which is including everything from the planning, from the processing, quality control, quality assurance, and quality improvement. And it's subjected to annual review. Which are specifics during the developing the QM plan is that we should define all the major challenges, achievements, and define recommendations for the next year. And it should be part of the QM assessment. For now, I will walk you through the QC essentials and how we implemented them in our practice. But before that, I just want to make sure that we all understand what is the difference between quality control and quality assurance. So quality control is determining if the product of the service meets the specification. And the pure definition of the quality control is that it's intended for the detection and correction of significant clerical and analytical error and unusual laboratory results in a timely manner. So basically this is telling us, are we okay? Can we continue our operations? Or we have a problem, so we have to stop and revisit and correct before we continue. For quality assurance is something different. So kindly note that quality assurance is not same as quality control, because the activities are not tied to actual performance of the process, and they are offering a retrospective review and analysis of the process, which is in state of control. So in this table here, you can see the comparative view of quality assurance and quality control, and what are the most important characteristics to remember is that quality assurance is a managing tool, a QC is a corrective tool. Quality assurance is always process oriented, and the QC is product oriented, let's say DNA, RNA, or viable cell oriented. Quality assurance will always offer a proactive strategy, so what we can do to prevent, but quality control will be reactive, so we identify the problem and now we have to act immediately. Quality assurance will gravitate toward prevention of the defect, and QC will go with detection of the defects. So there are some important things, and they are both equally important for successful quality management system. So when we come to the quality controls, there are a few things that we have to keep in mind after we understand what is the difference between quality assurance and quality control. So let's focus now about the QCs used in biobank laboratories. So QC is referring to systematic monitoring and evaluation, and it's measured and trying to identify and correct any issues or any errors. Quality control will tell us about accuracy, precision, and reliability in our results. We have three main types or three main categories of quality control. So the one will be pre-analytical, analytical, and post-analytical QC, depending on our process. Ideally, we should have a full circle to cover the QCs from the pre-analytical, so from the, for example, from the sample, handling, storage, and transport, then dive into analytical process, which will require quality control samples, calibration of equipment, validations of analytical methods, and post-analytical, which will be some kind of report, some kind of validations. I'm going to show you later on our examples. But before that, if you are on your way to select the best quality control for your scope, for your test, there are a few things that it should be considered before. So we have to know what type of assays are available and what we are going to perform. What about the matrix? That means the background of the quality control. What is its stability? What are the storage requirements? What is the cost? Because QCs can be extremely costly. We have to consider practicability, and we always have, thankfully, a big network and peer-reviewed and many published papers from where we can be creative and look for the best QC that we can implement in our lab and which can really fit our need. I'm going to show you now on the next slide how Qatar Biobank Lab has met those criteria. So when we think about the quality control types, there are some commercial quality controls which are usually purchased by accredited reference material manufacturers, and they are split to two main types. CRM, which is certified reference material, and RM, which is a reference material. So the difference between RM and CRM is about this certification part, because CRM will come with the measurement of certainty, and reference material, it will be just like coming with the conformity leaflet. It's extremely hard to find something for biobanking. If we think about plasma or serum or urine or saliva, it's very, very hard to find. But there is another way so that we can still comply. There is something called in-house developed QC. So we can develop our own QCs, and the only thing that is that we have to write them down, and of course we have to monitor them, we have to test them, and then later we have to monitor them. It's important that the in-house QC will, as any other QC, will comply to the two mandatory requirements, so that the value will be close to the range of expected results. So we will not compare pears to apples, so we will be in the range of our expected results, and that we will compare the same matrix. So for example, the same matrix like saliva, DNA from saliva or DNA from whole blood, it can have some differences. So that's why it's important to closely specify what kind of matrix is we going to use. And of course, once you enroll in the proficiency testing or interlaboratory comparisons, and once those PT samples will get their evaluation from the laboratory, you can always keep that sample and use it as your internal quality control, because it will give you a relevance and traceability to the correct result, to the correct value. Here are the examples of our in-house developed DNA QCs, how we are monitoring them. We are following Larry Jennings' charts, and also known as Westgard rules. And these are just a set of quality control rules, and with this we can detect errors and deviations. So it's a daily routine process, and it's been reviewed monthly for calculation of coefficients of variations. Once we developed our internal QC or we purchased into external QC, it's very important to keep a track and to keep a log of the sample types, when we start to use it, what is the expiry date, and if there is any QC violation, to document it, and of course to perform a corrective action after that. One of the examples of post-analytical QCs, what we implemented, it's a sample storage validation. So we agreed that we are going to check our inventory, that we will check the physical location with our information system location, the virtual one, and we will compare the location and we will compare the sample condition. We set up the criteria to be around 99%, and we are expecting that 1%, if we don't find it, that we will accept, but we are going to work on its improvement later on. The other thing is, which came from ISO biobanking. ISO biobanking certification is a quality report from where we had to assure to researchers that the biological specimens which are received, that they have been collected, processed, stored in accordance with established standards and protocols which are under our scope. So once implemented, quality control journey just starting. So that means that quality control needs to be monitored and evaluated in the timely manner. It's important to establish the policies and procedures. It's important to know what is going to be monitored and then to perform a regular review of the QC data. And this is, for example, what we get in our practice. So we are comparing and monitoring manual quantification for DNA, automation quantification for DNA over the years. And we are calculating the coefficient of variation for every month. So we will get a cumulative yearly one. And here we can see some trends that, for example, that we are getting better from year to year, that we are observing a higher difference between automated and manual process, which is, of course, expected. So this trend analysis based on your internal QC plan are highly important and highly effective later on, because it will just give also confidence to the lab staff and to give confidence to the management that we have things under our control and we are providing the best results we can. And another important part, as a part of the process management, is method validation or method verification. So why they are important? So the method validation or verification having the same outcome. So we need to make sure that our protocol or our method are successfully implemented in our practice. And for some methods, there are standard methods available, golden methods, golden standards. They are published and they are easily accessible and everyone can follow them. But also there are some methods which are not standard methods and they are also published, but every laboratory have to overcome some challenges for those implementation. So one of the most important things to remember when it comes to validation and verification is when we are doing them. So validation will always be tied to the non-standard methods or the methods which are modified. And verification is just a simple confirmation of the standard method or the method from the manufacturer. And there are specific cases when the validation has to be performed. So it's usually before the introduction to the method for the routine use. If new method is developed, any conditions which are changing, like instrument relocation, a software update, whenever the method is outside of their original scope, or during your QC review, you identify a shift or trend. So again, it will indicate that method needs to be validated. To perform a validation or verification, there are parameters that it has to be followed. So it will start from accuracy, precision, specificity, sensitivity, limit of detection, limit of quantification, linearity, and measurement of certainty. Here in Biobank, we are using a great Excel add-in. It's Abacus tool, and it is CE certified, and it's easily applied, and it's avoiding the human error during the stringent and complicated statistical analysis. For the accuracy, it's just important to use a good reference method, a reference material. So it can be CRM or RM or your PT standard, and this will be used as your nominal value. You can use student t-test, f-test, or anything which you can quantitatively express the accuracy in terms of trueness and bias. Accuracy is leading to repeatability. Precision is reproducibility. So that means that it's closeness of the agreement between the series of measurements which are obtained from multiple sampling at the same homogeneous sample under reproducibility conditions. So here it can be changed from the day, from the operator, from the instrument. So it's a very important part of the method validation to assess the coefficient of variation. So why accuracy and precision are so important? Because we can be, as you can see here, we can be accurate but not precise. So we can be precise but not accurate. And what we are aiming for is that we will be accurate and precise. And why is that? Because we want to avoid systematic errors and random errors. Systematic errors are tied to accuracy, and random errors are tied to the precision. So when we incorporate good precision and accuracy strategy, at the end we will reach to high precision and high accuracy in our laboratory tests. Another important parameter is linearity. It's mostly applied for the quantitative method, so it will talk about the range and dynamic range of the curve, and it will link on specificity and sensitivity, which can be analytical and clinical. Also, sensitivity can be analytical and clinical, and it will be expressed as a limit of detection, so the smallest amount of analyte which can be detected, and limit of quantification is the smallest amount of analyte that it can be quantified with 25% of standard of coefficient of variation. So all of those parameters that I already explained will end with the biggest one, which is called measurement uncertainty. And why is this? Because measurement uncertainty is, as you can see on this fishbone diagram, is obtained, calculated the contributions of accuracy and precision. So everything what we are using and everything related to the method is calculated statistically with the level of confidence 95%. We are using also the coverage factor of 2, and we can express our measurement uncertainty. So to get a reliable result, it's important to consider validation, traceability, and additionally the uncertainty. So for example, if researcher or anyone wants to know what is our measurement of error, we can give them the results of our measurement uncertainty. Here is a validation verification parameter summary, where we can see the types of errors, systematic, total, random error, how the performance of characteristics are fitting in with our validation parameters, and how we can quantitatively express any of those performance characteristics. So there is something to think about, that even if you are achieving 99% level of quality, it sounds great, but when you think, it means that we are still accepting 1% of error rate. So how does this reflect on our processes? How every laboratory needs really to find a way to see what is acceptable error rate for their own laboratory. Here I will just show you how we are following the workflow to perform validation and verification. So we will start with the request, where we will define our criteria. We will then develop a plan where we will put all the experiment design requirements, reagents, equipment, personnel, and we will end with the report where we will summarize our raw data and our results, and we will put a statement that is saying that this method is ready to use in routine work. Here you can see some of the examples. So this is our template for the request. So all the relevant parameters will be here. In plan, we will go in more details, we will specify, and we will always link this to any guideline like a TLSI or any peer-reviewed paper or white paper. And in the report, we have to take in consideration different things. So our scope, the characteristics of the required method, appointed operators, they should be trained and competent to perform this task and not to introduce additional bias and variable in your method, validation, types of the samples, how we prepare them, how we store them, what are the working environment conditions, what kind of equipment and accessories we used, and of course, the most important thing is a result analysis, and followed by conclusion, where we will put a comparative table, what we required, what we obtained, and what we are going to accept. With, as I said, very important statement at the end of every validation report that all these parameters were reviewed and approved, and they are ready for routine clinical and research testing. Thank you once again to give me this opportunity to present our quality journey. And now I'm ready and happy to answer any questions. Thank you so much. Good morning.
Thank you
Before I start, I just want
to the Qatar Biobank team for today's wonderful presentation. We've gone 12 minutes over time, and so we will take your questions and give it to the Qatar Biobank team to answer later on via email. But thank you once again, and we'll see you all at the next session next week. I believe if you haven't already registered for those sessions, there's a link that I will put in the chat once again.
I put it already, Chisom.
Yeah, just in case anyone has missed it.
Okay, yeah. So thank you so much, everyone. And thank you, Chisom, for moderating this session. And we'll see the participants next Tuesday, March the 7th.
All right. Thank you, everyone.
Thank you.
Thank you.